Welcome back. We're going to get started again for the next portions of our talk, our next four. My name is Chris, and this is Emily, and we're going to continue our talks on this really important topic. Our next speaker is Emma Barnes. Is going to be speaking on oiled vision, understanding fish's visual responses to oil spills through research and films. Thank you.
Okay. Right, thank you. Hi, I'm Emma. I'm going to be talking about sort of a mix of my research on vision testing, and then I'll be going into how I'm translating that research into an outreach project in the form of a short film. So as this session says, I'm going to be talking about the Deepwater Horizon spill, and after the oil spill, there was a range of research undertaken on a variety of different species, which includes red drum over on the right and also mahi-mahi, both of which have very large populations in the Gulf of Mexico and are very popular sports fish. And RNA sequencing studies, which were recently undertaken by Dr. Elvis Zhu, are suggesting negative but sublethal impacts on eye development and vision of these fish. And so the results of this study, along with results from other tests indicating that vision is being affected by oil, sparked a line of questioning that led us to investigate exactly how much oil is affecting visual development and visual processing. So unfortunately, you can't ask a fish to tell you if they can read an eye chart and what the lowest line of letters is on one of those charts. But luckily, methods have been previously developed to understand how fish are processing vision behaviorally. And so the concept that we chose for this research is referred to as flicker fusion. And this concept is pretty straightforward. If you've ever stared at a ceiling fan, initially you can see the individual blades spinning around, but as the fan speed increases, gradually the definition of each individual blade starts to blur, and you get this blurring effect around basically in a circle along the fan. So this is the concept of flicker fusion. And the way that we test this in fish is by putting them in this vision chamber. So you put the fish in the chamber, and then surrounding the chamber you have a sleeve that is lined with black and white stripes. And the fish will naturally choose a stripe and then begin to follow it around the chamber and just keep swimming until either the speed becomes too fast, like in the ceiling fan, or the light levels, or additionally the light levels are too low, so the fish also cannot see the stripes anymore. So my current project is looking at rod development, so low light vision, and preliminary studies are suggesting that at 3 days old, sheep's head minnows are able to not only see the stripes and track the stripes at full light levels, which is L5 in that series of photos flicking through at the bottom, but they can also see all the way down to L1, which is almost the lowest level that our optics reader can pick up. So that is really comparable to about 2-month-old sheep's head minnow as well, and adults. So that indicates that their rods are quite well developed at a very early age after hatch. And so the future upcoming steps of this project are to take mahi-mahi and red drum and expose them to oil embryonically, and after hatch, analyze and put them in that vision chamber to see how well they're able to track those stripes. And a developmental delay for the exposed fish is expected. Developmental delays are expected due to research that has previously been done in our lab, primarily by Jason Magnuson. You can see around that speaker picture, but I'll have the full information later. But Jason has done studies under full light conditions, under oiling, so he'll have a lot more information about the methods that we use to look at these tests. So there's a lot more involved, and Jason has also been doing a lot of histology, which you can see in the pictures there. There's those sheep's head minnow dissection histology slides after oil exposure compared to controls. And there are some significant differences that we're seeing. So if you would like more information, on Thursday, Jason is going to have a poster, and again, the information is there about mahi as well as sheep's head, and you can talk to either of us after this. So moving into the outreach component of my project, so not only is understanding how fish are affected by oil important, but also being able to communicate that into the greater area and people in the Gulf area. So I have a unique and exciting position of being able to directly translate the research that I'm doing also into an outreach project through film. I've had a very long-standing interest in film and photography and communicating concepts visually. You might have noticed I don't really have any text in my slides. I'm primarily communicating what I'm saying through photos and videos instead of having very much text. So vision in particular is a really good subject for outreach because it's really easy to understand. The majority of the population of humans, we use our vision all the time. We're highly dependent on our vision. And so looking at fish and developing an outreach project that focuses on vision and how fish vision is affected, because fish vision is relatively pretty similar to human vision, can develop an empathy with these species and really increase the interest in what happened after the spill. So the major aspect of communicating research is having an audience who is interested and is willing to listen. And I am in the Recover Consortium, which is a GOMRI, a Gulf of Mexico Research Initiative consortium, which has been really great. We have a really great outreach coordinator, Dan DeNicola, who has run a lot of outreach projects along with the rest of Recover. And all of those outreach projects are going to be talked about by Martin Grissel at the 11 a.m. talk in this session. So he'll be giving a lot more information about that. But with Recover, I'm pulling in some audience through Recover, which has been really great. But I've also developed my own social media accounts to build an audience further. So right now, I have an Instagram account, and I'm looking to expand further. But the Instagram has been really interesting. I've only been running it for a couple of months, but so far I'm getting a really wide diversity of audience. So not only am I getting interest from science students and filmmakers, I'm posting both a mix of filming background for my film, but also how my research is progressing. But I also see likes and follows from environmental and conservation organizations, a lot of fishermen, which has been cool, a lot of sports fishing groups that take people out fishing, and also even a couple of drilling companies that have posted a few photos of oil rigs, and I'm getting interest from just a huge variety of audiences, all of whom could potentially be impacted by the spill, but in a variety of different ways. So it's been really nice to see that diversity. So alongside working on building an audience, once I have that audience sort of interested, I need to keep them interested and really focus on encouraging an empathetic connection between the audience and the fish that I'm going to be talking about. So the film is currently underway. It's not yet completed. I do have a trailer and a couple of slides. And I'm working on bringing in some interviews that are non-science or non-academia professions. So the first is Tony Ludovico. He's taken all of these really great photos of drum and mahi that I've had throughout my PowerPoint. He is a marine photographer, so he really gets in the water with these fish. He sees them in their natural habitats. He's interacting with them. And so he's really able to see their personalities. Are they cautious? Are they curious or aggressive? How do they interact with their environment, and how do they move through the water? And how do they see the world, essentially? So I'll be talking to him to really bring in sort of a curiosity for the audience towards these animals. And then my next interview that I'm working on is with an ophthalmologist, an eye doctor, as you can see with that picture of one of those crazy looking machines. But the ophthalmologist I'm going to have talk about human vision, how human eyes work, because it is relatively similar to fish vision, how human vision is tested and how that compares to how we are able to test fish vision, and also what kind of problems that developmentally or through age people can have with their vision. So I'm just going to briefly go through the film layout that I have set up right now, and then I have a short trailer to show after that. So initially, I want to open with just a basic background on the Gulf of Mexico. What kind of environments are there? What species live there? I've got a series of clips from a fishing tournament that I attended, and that really gives an understanding of what kind of variety of species are living in the Gulf. We'll be bringing in Tony Ludovico to talk about how these fish are interacting with their environment again, and focusing on red drum in more coastal environments, and also mahi-mahi in the more pelagic environments. And then I'll be introducing the spill and talking a lot about embryonic development after oil exposure, because that's where we see a lot of the lethal and sublethal effects of oiling, and comparing and contrasting that to human vision. So bringing in the eye doctor that I've been talking to really have that comparison between fish and humans. And I'll be talking also a little bit about the research that I've been doing. So that blue image in the bottom row there is the vision chamber under infrared light, which is one of the methods we use for a control. And then finally, I'll be finishing the film. I'm estimating it to be about 20 minutes, talking about the future of the Gulf, what kind of prospect oiled fish have after exposure, and hopefully ending on a positive note for the future. All right, and without further ado, this is my trailer, so hopefully you can hear the music. And with that, I'd like to thank the Gulf of Mexico Research Initiative and also the Recover team, along with Tony Ludovico. And again, if you'd like more information on the research aspect of this, please go and see Jason Magnuson's poster on Thursday. And if you'd like to follow me for updates, my Instagram is Fish and Oil Spills. So, thank you.
We've got a good amount of time for questions, about 4 minutes or so. Does anybody have any questions?
Hi, thank you. Josh Butler from ExxonMobil. I know that research is ongoing for this currently. Is there any preliminary research that you might be able to refer to that suggests oiled vision may be one of the more sensitive physiological endpoints that are researched at the current time?
Yeah, so the RNA sequencing study done by Elvis Zhu, who is also in our consortium, he found that some of the primary targets of oil was both in cardiac effects, which our group has studied a lot, but another less studied, there's a lot of targets on the vision and eye development as well. So definitely going off of that, as well as there has been previous studies after oiling that there are definitely effects on vision, aside from the cardiac effects as well.
Okay, thanks.
Hi, I'm Jordan Kozel from Duke. So the vision assay that you were using also relies on the fish's ability to move, and since oil also affects bioenergetics and swim performance, how can you really tease out whether their ability to follow a bar is a vision effect versus a bioenergetic effect?
Yeah, so this is definitely something that we're initially just wanting to look at sort of basic vision, but definitely movement can play a role in that. And there are tests that you can do where you fix the fish so it's stationary and you just look at the movement of the eye as it follows the stripes. So that's definitely something that we could look at in the future as well.
Thank you.
Any other questions?
All right. Well, thank you very much.